forget how much easier this is when things like this happen. Anyway, it is my great privilege to introduce Bikash Pattnaik coming to us to give us a lecture today on some of his very interesting work in which he's been using genomic approaches to try and restore function. Pattnaik did his PhD in biophysics at Delhi University and came to do a postdoc for Brett Hughes, right? And Brett Hughes was trained by part of the Sheldon Miller and Roy Steinberg electrophysiology of the RPE. And he is part of a very exclusive group in that he's patch clamped the RPE cells. And so there's not very many of us of them in the world. And so there's a definite bond with people who've actually struggled with the patch clamping of the RPE cells. And this allows a real intuitive sense of function and physiology and how these cells are working. The NEI doesn't quite see the value of patch clamping RPE cells that we do. And one of the things that it's been fabulous to watch Bikash kind of develop is to take his skills at being able to understand the functional aspects of the electrophysiology, both on a patch clamping level, but also as clinical director of the visual electrophysiology, and fix things. And now we need, it's not just enough to know how things work, you've got to go in there and repair them and fix them and cure blindness. And he's had, I think, a very successful project in going back and doing splice editing and trying to go back and adenine base editing to repair single point mutations. But it's also, I think, a story of perseverance, how very often developing the model is harder than finding the cure. And I think as a patch clamper, you've done very well for a patch clamper. I think it's very, very nice to see how, curing things is better than patching them or whatever. So without any further ado, I will let Bikash tell you about some of his interesting work. Thank you very much.
Thank you, Claire. In fact, Claire and I, we have met over this point, the exact point that she made, that how come I can't have people read my grant, but then they are reading your grant and funding. That's what she complains about. But then, yes. So yeah, I'm a hardcore electrophysiologist. That's how I got trained, especially in biophysics. But as you see, my talk would be mostly using genomic medicine to see, how we can correct these errors and then let the ions do the talk. Thanks, Claire, for inviting me to give this talk. But then, so I have been to this campus several times before this, but not as an invited guest. My tie to this campus is also my son, who just graduated. He's sitting back there. So this is my family, and I say we are all things considered. So my son, who is here at Penn, he just graduated. My daughter, she's at UW. She's also doing some neuroscience, and then myself there with my wife. So with that, I don't have a financial conflict for the talk that I'll be giving, but then some of the patents around the KCNJ13 gene therapy has been licensed to a Hubble Therapeutics company that it's a spin-off from UW, and then they are taking it to clinic. So the learning objectives for today will be, I will touch on equitable access to healthcare. We'll discuss the molecular mechanisms of ion channelopathy and how I have used it for testing novel therapies, and then expanding precision medicine in this era of IRD. And then my goal would be by end, I would have proposed a therapeutic menu card, especially for LCS16, and that's what I study. So this is a story which was published by UW, which basically addresses people living with rare diseases have long faced a common challenge of equitable access to health. And that's where we teamed up. So this is Dave Gam, myself, Sarah Gong, and then Chris Saha. So we have been teaming up to develop this gene editing therapy to treat blindness. So, patient that has made my life or that has changed my life or my perspective on electrophysiology. I'll go over the medical history of this patient. So this patient, when he was 10-year-old, he was first seen at Cole Eye Institute by Elias Trabulsi, where there were some clinical phenotypes which was noticed, like nystagmus, strabismus, photophobia, cataract. Visual acuity was 2200 and 2400, and then pigmentary retinopathy. Then we have been seeing this patient. So we saw the patient in clinic in 2016 and then in 2020. So when we did, these are all the clinical diagnostics. So this is a fundus picture. So this is a normal retina with normally developed healthy blood vessels around the fovea. And this is our patient when we saw the patient for the first time. So you can see around the macula, there is some pigmentation. And then most important, you see these are the phenotypes that we see, classical phenotypes in these patients is the attenuated retinal vessels, and then early pigmentary alterations, which attest to mottling of the RPE cells. And then when OCT was looked at, this is a normal OCT from a healthy subject. So you can see all the defined retinal layers, which are preserved in this image with hyperfluorescence layers. And when we looked at the patient's OCT, so you can see loss of both easy band and then also which reflect to RP disruption here in the posterior retina. Then the third criteria is electrophysiology. So this is a normal, healthy control eye, 2-year-old. So you can see these are dark adapted ERG, and then these are the cone responses. So they have normal B wave here at the lowest intensity. And then if you increase the intensity of the light, you see A wave, B wave, and then OPs. Then these are the photopic responses of single flash and then also flicker responses. Whereas when we saw this patient, so the patient had reduced amplitude, not eliminated completely flat ERG, but ERG was reduced with normal latency. Then the blood samples were sent out for genetic testing, and then it was found out that the patient has a homozygous mutation in the KCNJ13 gene for a G to A transition at position 158, which is basically introducing a nonsense codon, which is tryptophan at position 53 in the codon. And so then what we did is at a follow-up exam, when the patient was 21-year-old, you can see that this is now a flat ERG. So there is a progressive degeneration in the photoreceptors. And then also more pigmentation here, plus also this is more degeneration in the, or more dystrophic outer retina here. So there is undetectable ERG in this patient. So then what we did is, because I was studying this channel from a physiological standpoint in the retina, where these channels are present in the RPE cells. So here, if you see, this is a retina. So these are photoreceptors here, and then these are the RPE cells. So this is the expression of KR 7.1 along with ezrin, which is a molecular marker of RPE apical processes. So if you see, they both co-stain, so or they have co-presence here. So the KR 7.1 happen to be a monogenic disease, which is only present in the RPE cells at the apical processes. So that was the starting point of me thinking that, okay, since this is only one cell type that I have to treat, so probably this is a really good model to test some therapy using the models that we have. So then, so why ion channels are important in retina physiology? So you all know about this eye and then retina. So these are the RPE cells, photoreceptors, bipolar cells, and then ganglion cells. So starting with phototransduction, ion channels are involved. And then most importantly, that the phenotype of the ion channel, which is basically a sum response that you see in the ERG. So photoreceptor A wave is a hyperpolarization of the photoreceptors. Then the bipolar cell response is depolarization. And then this is a human patient recording showing C wave, so which is again coming from the RPE cells function. So if I put this into context for ion channels, so what ion channels do is these are proteins on the membrane which permits ion to go in and out. So when ions move in here, so if you imagine, so a ion, basically a charge is moving, so it generates current or electricity. So, and then if you close this channel, so that gives the resistance. So basically we can plot this into a normal equation, which is Ohm's law. So the voltage will be across the membrane. Current is when the charge is flowing, and resistance is when whether the channel is open or closed. So if I put this into context, so V is equal to IR, which is the Ohm's law. So we can basically find out the relationship when the channel is working and when the channel is not functional. So it can be mimicked here. So if you provide more resistance, then the ion flow will be inhibited. So that's how we can plot this by a simple equation, V is equal to IR. And then the other thing is about, because these are potassium channels. So if you look at potassium homeostasis, there are clearly 3 potassium distribution homeostasis within the retina. So there is one at the glial cell level. There is one at the across the Muller glia, and then there is one here in the subretinal space. So basically, if you have light activated, then there is a drop in the potassium concentration here, and then we have the KIR channels in the RPE cells to facilitate ionic efflux, so that the potassium homeostasis is maintained. It's got back. And then, so that's why we started with this hypothesis that when these channels are non-functional, that's how they cause the disease, because your ionic homeostasis around the photoreceptor outer segment is altered. So that could be manipulating the physiology, altering the ERG of these patients. So to test that, what we did is we used this UW, University of Wisconsin is known for the stem cell technology. They were earlier on to do that. So what we did is we got consent from the patient to donate us biological samples. So we got a skin biopsy from the patient, and then we generated these iPSC cells. So these are the diseased iPSC cells. And then also we were lucky to convince one of the family members, which is a normal healthy subject, to also donate biological sample. So we could have a epigenetically controlled iPSC cells from the same family. So when we looked at, basically you can see that this subject is this healthy subject is a carrier, because you have both A and G here. Whereas for the patient, it is a TAG. So it's a TGG to TAG mutation, which is the nonsense codon here. If you look at the expression of the molecular markers, they are normal between the patient's line and then the healthy iPSC cells. But when you look at the protein expression, so KIR 7.1, because it is truncated, it cannot be detected by an antibody, which detects basically the C terminal of the protein. So that's when we confirmed that, okay, so we now have at least a molecular phenotype in these cells. So protein expression is altered. So then, as I said, that we electrophysiologists, we are interested in these plots, which are called IV plot, or a voltage current relationship. So then we recorded from these iPSC cells, RP cells. So these are individual dissociated cells. If you see, these are polarized, and that was very important for us, because unless these cells are polarized, they don't have the apical processes, the KIR 7.1 will not express. So initially I struggled for a while to find out the KIR 7.1 current, because it won't express in any iPSC RP unless you mature them to develop all these apical processes. So then when we tested, so wild type has this current, and then the mutant is in red. So you can see that the mutation causes the cells to be depolarized here. So if you look at the membrane potential, that was the first thing. Then the other characteristics is these channels, the KIR 7.1, they can be activated by a simple ion, which is rubidium. So if I replace potassium with rubidium, so rubidium has a higher permeability through this channel, so it enhances the current. So that also showed us that for the wild type, I could enhance the current, but the mutant was unaltered. So with the rubidium, there was no change in the function of this channel. So that was also a clinical phenotype that we saw in these cells, which confirmed that not only the protein doesn't express in these disease cells, but it is non-functional here. So then that made us clear that if we tested a therapy and we could rescue some of this function or expression, then we know that this therapy is working in these iPSC cells, patient iPSC cells. So what's KCNJ13? So KCNJ13 has got multiple pathogenic variants. In fact, I mean, this is one we discovered. We are also now several others which are coming. We are writing this paper, L144P patients we have seen. There is also multiple families with 114. So this is basically a 2 exon gene. It's a very small gene, 2 exon gene. It gets translated to a 2 transmembrane protein with a small N terminal and a large C terminal. And then this is the selectivity loop for potassium. So the question that next I ask is, so this is the premature stop codon we have at position 53. So it is clear that with this, we are not having a full length protein product. So that's why it is non-functional. But how come some of these mutations, that's why it's a recessive disease. But then this one, L114P, is a autosomal dominant form of the disease. It's not a LCA, it's snowflake vitreoretinal degeneration. So we got interested in this also. Can we identify the molecular mechanism? What causes autosomal recessive? What causes autosomal dominant? So that's why we looked at this mutation here, this specific mutation 114, which is in the ion selectivity of this channel. So to look at that, so this is the SVD fundus, where you look, you see this snowflake like deposition here, and then the mutation is here, L114P. So basically, if you look at, so at this position for the KIR family, either it's an isoleucine or leucine, which is conserved. But then by putting a proline here, so it has the phenotype. So that's why we compared here, and then this was done by a training in the lab, Feza, who was visiting us from Turkey. So then when we did this in a heterologous expression, so what we tried to find is why it causes a autosomal dominant phenotype. So we did wild type here, it expresses on the membrane. When we looked at co-expression of the wild type with the mutant, it's also present on the membrane. And then when you have mutant, it is present on the membrane, it's also present in the cytoplasmic location. So if you look at the Pearson's correlation, they are present in the membrane microdomain. So it is part of the functional channel, but it is altering the function of the channel, being part of that. So that's when we looked at electrophysiology. So here, this is wild type in blue, and then in red is mutant, 2 copies of the mutant, or it's all mutant. When we expressed 1 copy of the wild type, 1 copy of the mutant, you can see how drastically it changes the phenotype of this channel. Okay, so here this is a classic representation of the inwardly rectifying. It bends inward, that's why it is called inwardly rectifying channel. And then in these cells where either expressing the mutant alone or mutant along with the wild type, it disrupts the function of the wild type, making it autosomal dominant. But when we looked at rubidium as a charge carrier, this work was done by NS, who is a postdoc in the lab. So if you look at here, charge carrier, rubidium is still carried through the heterologous channel, both wild type and mutant. So then we started to hypothesizing, is this only non-functional for potassium, but it is still functional for rubidium? Maybe that's the case. So that's where we looked at here. So this experiment, when we did is, this is wild type alone, and then we changed the ratio of wild type to the mutant. So what we hypothesize here, so there is a size difference between potassium and rubidium. So for a wild type, both rubidium and potassium can go through this channel. But when we have a mixture of wild type and then the mutant, then here only rubidium can go through. And then you will come to a situation where you have only mutant channel and then rubidium doesn't. But for potassium, for all practical purpose, this is a non-functional channel. And that's how it generates the autosomal dominant phenotype, which is also shown here. I can summarize all this here in this one. So in red is the rubidium permeability of this channel, and in black is the potassium. So for potassium, if I drop from wild type to any ratio, it's basically flat. It doesn't function. Whereas for rubidium, it shows a progressive decrease in the current. So that's how it generates a autosomal dominant, because it interferes with the function of the wild type component of the channel. So then we try to test why it does that. Does it also do for other KIR channel? So KIR 2.1 has an isoleucine in place of leucine. When we tested KIR 2.1, we switched it to leucine. It functions normally, but when we change to proline, it's again the same thing that we saw for the KIR 7.1. So now we know that, this is a conserved location where it cannot be changed, or it will have even for KI 2.1. If in future somebody finds out that there is a mutation here, isolation to proline, then we know that it will have a phenotype in the patient. So we looked at the structure of this protein. So if you look at this structure, so this is normal leucine wild type channel. And if we replace that with proline, so we did a protein modeling, and then it changes the kink, or it alters the pore structure. So potassium can no longer be flowing through this selectivity pore of this channel. So in summary, so we not only found out the mechanism, but we have the tools to develop therapy, and that's where we got in. So let's see if we can test some of the therapeutic tools, because we have possibility of correcting at the genomic level here by gene augmentation or gene editing at the nucleus. Or there are also, because this is a nonsense mutation, we can also study read-through, whether we can manipulate the protein translation and we can get a full-length protein product. But this is a channel, so we can show not only we are getting a full-length protein product, but we are getting a functional protein here. So to do the gene augmentation, then what we did is we took a healthy copy of the human open reading frame, and then we packaged it to AAV. So initially we tested it using a lenti for delivery to the iPSC RPE cells. So these are our LCA 16 iPSC RPE, which normally doesn't express Ki at 7.1. But when we did gene augmentation, you can see that Ki 7.1 is present. Not only they are present in the apical processes of the RPE cells. When we looked at their functionality in this, you can see that now this was non-functional here, but after the gene therapy, they are fully functional. Even they are doing better than the wild type cells. For example, here current we saw in the lentiviral transduced cells, it was increased multifold. But when you look at the membrane potential, so it was depolarized for the mutant cell, but now we got to the normal hyperpolarizing RPE cell level there. So gene correction, this was reported, this was done by Pawan Sahi and then reported in this paper. So this all works in the iPSC RP because we can just drop in the lenti, but does it also work in vivo? That's when we tried to first test it in the iPSC RP, so compare it with the isogenic control. So here, this is the mutant line. This is the isogenic line where we corrected it. And then this is the treated one. This is our commercial hub 101, which we call, which has the AAV variation with a VMD2 promoter to drive the delivery of the gene of interest. So this is where you see the functionality. So they are not activated by rubidium, nor they are sensitive to the inhibitor. Whereas here, this is the isogenic line. You can see we have increased the current multifold here, and then same is with after the transduction. So here we saw that either the gene editing or the gene augmentation will be successful. That's when we try to create an animal model to test this. But, to our surprise, these animals which were homozygous, they will not survive day one, they'll die. So we used a conditional knockout here. So here, this is wild type. In the conditional knockout, because we were using a VMD2 promoter, so we will see multiple phenotype. In some category, we saw reduced A wave, B wave, and then C wave was eliminated. And then in some where there is a severe degeneration, then all retinal ERG waveforms were gone. So our logic was, if we treated this one where there is no C wave, and then because they have all the retinal layers with gene therapy, maybe we will be successful. And that's exactly what we did. So when we tested for our delivery using a reporter, so you can see that they are expressed in the RPE cells. This is safety testing in normal C57. So you see the C wave is about 460 microvolt, which is the RPE cell function. And then this is after the gene therapy delivery. So there was no change in the C wave amplitude here. So that means it's safe. Then when we test the therapeutic outcome, so this is a progressive increase in the C wave after we delivered the gene therapy product here. So this was completely gone here, but 1 week you could say a little bit of C wave rescue, but then over time as you see here, which I have averaged. So non-treated cells, they stayed here, non-treated animals, they stayed here, no C wave, whereas treated, either we are using a universal promoter EF1 or VMD2, they rescued the C wave in these animals. And that's what we are taking to the clinic through Hubble Therapeutics. Now, I wanted to talk about our effort in genome editing, so which was published recently in JCI. So where our goal was to package the genome editor in a non-viral delivery system, deliver it to the eye, and then at least if we get a functional channel here, then we should see some rescue in the function. So with that, what we did is, so we collaborated with David Liu's group to get the base editor from them. And then Sarah Gong and Alex, they helped us to package them into lipid nanoparticles. And then Chris Saha was developing this gene therapy. So I got some guidance from genome editing. So I got some guidance from him. So basically our idea is how we precisely fix this TAG to TGG so we can get a functional protein made. So this is intro of the base editor. So either depending on whether you want to change the A base editor or C base editor. So they are connected to the Cas9. And then they enzymatically will change the adenosine to inosine so that you can get a correction here. And that's exactly what we used in our case to use the ABE. So what we did is, so this is our vision in collaboration with David Gam and then our group. This is the NIH consortium that we have. So our goal was to use either patient fibroblast or patient iPSC cells to deliver these through silicon nanoparticles and then do sequencing or electrophysiology to find out that there is a functional rescue in the channel. So this is the, so initially when we tried it in the cells, so this was our sequencing readout. So we got like almost like close to 50% correction of the editing here for the TAG. So then genome editing poses a different challenge to electrophysiology because electrophysiology, we know it's very low throughput. We are going one cell after that. But then with genome editor, you will have multiple outcomes. Not all cells will be edited the same way. Plus you will have off-target response. So how do we handle that? So to do that, what we did is we took help of this, which is an automated batch system, because we had already established an assay for our system. We can use activator, inhibitor. So if we can reproduce that in an automated system, we can, in one experiment, we can test multiple cells, and then we will know the outcome in this condition. So what we did here is this is a Sophian system where you can record from 48 cells in one experiment. So this is on the right is the Hubble system. Cell in this microscope, the cell has to be identified, and then you do go one cell after. iPSC cells, it was challenging because, you know, I mean, we spend so much resources in making these iPSC cells, growing them over more than 200 days. And then if we can get to one cell, then we have lost all our resources. So that's where we wanted to do this. So with this system, you can see it's all robotics driven. So I can patch onto 48 cells in this. So it has got 48 amplifiers built into the system. The cells are automatically picked up by the robot. It will be dropped here. So I can get results from 48 cells in one experiment. So doing that, here you see, so this is our panel. So these are recording from the disease cells. So with rubidium, you can see there is no current ever, whatever you can see here. These are all non-functional channel. But after we corrected, so you can see here, there is at least 1, 2, 3, 4, 5, 6, 7, you know, I mean, these are almost 8 and 9 cells here, which showed some rubidium current. There are also non-functional channels here. For example, this cell here, but more or less we can look at the spectrum or the spread of the edited cells versus the non-edited cells in one experiment. So these are the electrophysiological recordings. So this is a wild type, which is responding to rubidium. And then these are the non-functional, which I showed you earlier. But then after the base editor, so we do get different rates of correction here. But then, they result in a functional channel EPDs. When we compared 65 wild type cells to the 65 base edited cells, we didn't see any difference in the current amplitude, nor their potentiation by rubidium. So this shows that the genome editing was effective. But then, how do we test it in vivo? I said that we didn't have an animal model to do that. So what we did is we used the animal model that we had, which is a head. So first we thought that, okay, because the heads, they express one wild type and then one mutant allele. So somehow if we use genome editing to disrupt this wild type, then we will have a mouse model with only the mutant present there, or the disease allele present there. And that's exactly what we did. So we CRISPR edited the wild type to get a mouse model with one allele left, which is the PAG here. And then we can base edit to show whether we get a functional rescue in this mouse model or not. So using that, so here is a example. So this is normal C wave. So after we did CRISPR genome editing, so the C wave goes down here and then it stays there. You can see there is also degeneration, starting to degeneration here in the retina. Whereas after we deliver the base editing, the cells look, or the retina looks healthier, and then there is rescue in the C wave compared to what we did after the genome editing here. And then this is the average response. So on and all, so we did see that the C wave was rescued after base editing in this mouse model that we did. So, and then this is the delivery. This proves that, okay, we can deliver, we can dose it. And then we did a multifocal in the mouse model to show why we had only partial rescue in C wave, but not fully rescue. Because this has to do with where exactly we are delivering. So anywhere we deliver the base editor, that's where we see the rescue in the ERG, but anywhere there is no editor present, then we don't see that. So that's why we got a partial rescue, but not a full rescue in phenotype. So those are all good. Okay, so I showed you that we can now correct this at the genomic level, or we can do gene therapy. But then how do we take this to address, the genes which are large, they cannot be packaged, or to develop a therapy which is not gene specific, but mutation specific? That's where our model was very useful, because this is a nonsense mutation. So all you do is, so as far as the gene of interest is there, so gene is there, it gets transcribed, but you have a mRNA which has the mutation here. And then this mRNA doesn't get translated, so you get a truncated protein. So we thought that because there is this read-through compounds, drugs which are available through PTC or ILUX, then if we use these compounds, maybe somehow we can do a bypass this translation termination to make a full-length protein product. And then we had the power, because we were doing electrophysiology, so we would be able to say whether it is a functional channel or not. But then we failed when we did the drug testing, because what was happening is the drug is inserting an amino acid, but it's not necessarily inserting a wild type amino acid. So it's still a missense. So that's when what we did is, now we have developed a newer technology, which is a tRNA therapy. So what it is, so this is in collaboration with Chris Harron from Iowa. So what it is if you look at a normal healthy mRNA, so you have UGG here in our KIH 7.1. So it goes through normal translation, you get a full-length protein. But when you have this termination, premature termination codon, UAG, so this basically terminates it prematurely, because you have the eukaryotic release factor, it recognizes the nonsense codon, you get a truncated protein here, which doesn't even have a transmembrane, so it doesn't get to the membrane, it is non-functional. But with this tRNA, or the drugs, what it does is they will compete for the binding of the eukaryotic release factor to the same codon, so that at least you can get a full-length protein made there. So, and then also, as I said, so this can target multiple ion channels, especially since that's what my focus is. So I can focus on both photoreceptor, RP, and then multiple different ion channels in the eye. From the subretinal space, I can target both RP as well as the photoreceptors. So to talk about the transfer RNA. So RNA, tRNA is the oldest known structural RNA that we know about historically. And it is almost like 10 to 15% of total cellular RNA is tRNA. And it's a very small gene. It's only 60 to 90 nucleotide. And then it has an unusual base pairing, as you see here, that gives the clover-leaf pattern of the tRNA. But there are most importantly two specific sites that I want to talk about. So one is here, which is the amino acid acceptor arm, where the amino acid basically get tagged to the tRNA. And then this is the anticodon, where this is recognized by the mRNA. So what we thought is if we take the tRNA with a normal amino acid, and then we alter the anticodon, then it will recognize the nonsense mutation of our choice. So here, if you see, so our normal healthy is UGG. So the tRNA is a tryptophan tRNA, and then the anticodon is ACC. But then the mutation is UAG, which is a termination codon. So if we alter this ACC to AUC, so it will be recognized by the nonsense mutation and insert, most importantly, a wild type tryptophan into the sequence. And then electrophysiologically, we'll have a normally functioning channel. And then historically, we have only a few nonsense mutations, arginine and tryptophan, and then glycine. So we have to only target specific sequences to deliver this tRNA. So here is an example of our control cells, iPSC cells. You see KIR 7.1 is expressed on the membrane in the apical side. These are our disease, which I showed you earlier. There is no KIR 7.1 made. This is after the tRNA therapy. So this tRNA was delivered using a viral. We used a helper-dependent adenovirus, and you see KIR 7.1 is made just by delivering a UAG tRNA. Not only that, I mean, it's almost like present in all cells here. And then electrophysiologically also, we could show that now these channels are functioning normally. Not only the channel protein is made, but the protein is functioning normally using this tRNA. So we tested it in our mouse model. So here, this is localized delivery again. And then this is knockdown. This is control. This is knockdown. And then this is after treating with the tRNA. So it shows a similar potency as the base editor, which is when we are now working on optimizing its delivery so that we can deliver tRNA more efficiently to the whole retina. So we get a better rescue of the function. As I said, this is a gene agnostic. So here is an example. So that was for KIR 7.1. This is an example for BEST disease. So this is a known mutation for the best one, which is arginine 200 at position 200 position, which is a termination codon here. So the best is in red. So this is healthy best expression. This is the mutant best, which doesn't make a channel. And this is after 3 different formulations of our tRNA. You can see in all 3 formulations, we do see a membrane localization of a normally healthy best channel. Not only that, when we did electrophysiology, so this is the mutant, which is not making a calcium activated chloride current. This is a wild type, and this is with 3 different formulations of our tRNA. We see a varied or a spectrum of recovery of the function of this channel in these cells, which right now we have also tested in iPSC cells. Here you see that this is the mutant channel, which doesn't make the bestrophin protein. This is wild type, and this is after the tRNA therapy. Both these formulations, they make a 68 kDa best one protein after rescue. So, and again, I mean, we have also filed a patent about its long-term delivery. So you can either use viral delivery, or here we are promoting a technology which is called mini circle. So it's basically getting rid of the bacterial portion of the plasmid and only delivering the tRNA, because it is so small that we can deliver it as a mini circle. And with mini circle, the advantage is we see a long-term expression in our RP cells in both animal models as well as in the iPSC cells. So as I promised in my initial propositions, that I will be able to provide a menu card to my patients. And this is exactly what we have done. So using both manipulating the transcription label or translation label, now I can show that genome editing is effective. Read-through drugs to some extent will have a phenotypical outcome, but then most important is with the engineered tRNA and then gene augmentation probably will get a better rescue of function if we wanted to treat these conditions. So why this is addressing health disparity? Because, I mean, I have had to deal with some of the patients. When the patients come into clinic, they get diagnosed with a disease, but then they are, now it's up to the patient whether they want to follow up with the clinic because they are so disappointed with this disease, they don't know what to do, right? So that's when it comes to the provider, whether to give them some guidance about what's out there and what could be a possibility, whether somebody in the world is working on that gene, or there could be a therapy available to that. Because, health equity is not, I mean, we see this health equity every time because health equity means that everyone has what they need to be healthy and that no one experiences unjust barriers that limit their health, and especially for ultra rare or rare to ultra rare, because we don't know how to treat those. And then they face the most barrier in here. So we did this collaborative study as I presented to present that, okay, we have got gene editing, gene therapy, the engineered tRNA, and then the read-through drugs. So we are bringing hope in focus for LCA16 gene therapy. As Dr. Martin Luther says, of all forms of inequality in this world, injustice in healthcare is the most shocking and inhuman. So this is also a story which was published by UW. Basically, this gives me my timeline at UW. So I came to UW in 2008, and then in 2009, we had published this iPSC, RPE, and photoreceptors as stem cell model for modeling disease. And then in 2013, we were already showing disease in a dish by looking at best disease because, there is debate whether best is a calcium activated chloride channel or not using different animal models. It has failed, but here we could show in 2013 using the iPSC cells that it does form a calcium activated chloride channel. 2015, we had a therapy for LCA16 using again the stem cell technology. 2019 and 2020, we had gene therapy for both LCA16 as well as BEST. And in 2023, we had shown that genome editing is effective to treat this disease. So I would like to thank the members of the lab who drive this work. And then I have had several collaborators, both within UW and also outside UW. And I have received funds from NEI and then also from foundations to do all this research. I'm happy to take questions.
And also the tRNAs that permit read-through the stop codon. How do you make that specific to the gene with that you want to target and not just getting read-through of all the stop codons?
So I can talk about the tRNA because that's the one we did the most, because since we developed that tRNA, so what we did is, so we tested this tRNA against the normal ribosomal read-through. So basically, we were reading at the termination codon to see how this tRNA acts on the normal translation codon versus the premature stop codon. So it has to, we believe that it has to do with the poly A, which comes in the normal stop codon. Okay, how the normal stop codon is recognized versus how the premature stop codon is recognized. Plus, the other thing is, if you look at the eukaryotic system, in almost like 95% of the cases, TAA is the normal termination codon. Very few with the TAG or TGA would be the codon which will be preferred for the termination. So that's why we got lucky. So both using the ribosomal study as well as here. Whereas with the PTC, since we were involved, what we did is, so when we tried to treat with PTC, like we have these Ilux compounds, we have the PTC 124, PTC, 84. So we tried even from Richard Gatti, I got some of his RC compounds and we tested it all. So we had a mixed response. Like for some of these drugs, we will see there is a therapeutic outcome, not as good as the gene therapy. So that's when what we did is we hypothesized that it's not inserting a wild type amino acid. Then we went ahead and looked at the codon. What are the possible amino acids it could be recognizing? Because let's say you recognize only 2 oval positions, in the codon. So we got into all these possible amino acids and then did a site-directed mutagenesis to find out which one is giving us phenotype and which one is not. To our surprise, we found out that most of the amino acids will not work. It's only either a wild type or very close to the wild type amino acid. It's giving us a normal function. So that's when we abandoned the PTC approach, not the therapeutic drugs. And then we went to the tRNA, which is very specific because we know that we are inserting a wild type amino acid into the sequence.
So what same cells for the gene editing and also for the tRNA?
Yeah, so for the tRNA, so for the mini circles, right now we are using LNPs because, they are proven for therapeutic uses. It will be easier for us for translation. That's why we are switching to LNPs instead of the SNPs that we reported earlier. And also with LNPs, we have got more flexibility to get commercial sources of LNP and then compare with the commercial sources. Because one thing that didn't work for us is after we reported the SNPs, we made several other SNPs and then there was a batch to batch variation in the yield. Okay, which we went back and then, there was probably 2-year study that Sarah Gong did to find out what chemical is causing this, and they couldn't make out. That's when we switched to LNP because of all the known results. Because Moderna uses for your COVID. Yeah, no, not for retina. Exactly. But the thing is with the SNPs, what we did is when we did the silicon nanoparticles, we could change or we could modify the surface of the nanoparticles to be recognized by either RP cells or photoreceptors, even if they are delivered to the subretinal space. So that's exactly the approach we are also taking from LNPs. Although LNP has not been delivered for retina, we are surface modifying the LNPs to be targeted to either RP cells or to the photoreceptors.
Which do you think have expression of the gene? Are they comparable?
No, so tRNA is much better. Yeah, tRNA is much better, very specific, because with editing, what happens is, it has to do with the editing efficiency. Again, editing efficiency also varies from, one prep to another one. So like, we reported that we have seen up to 40%, but then, there are experiments where we are only seeing 20% or less editing. And then, but the industry says that if you get more than 10%, you are successful. But, not for us. We are not seeing a functional rescue with 10%. Right. So that's why we have to up our game, but then we don't see consistently. Whereas with the tRNA, as I showed, there's multiple preparations we have tried against multiple genes, and then everywhere we are seeing that there is a functional outcome. So that's why we are very positive about the tRNA, and that's what we are driving now.
You've done
potassium. We have done with potassium. We have done with other ions, also positive ions. But I think this channel is very unique compared to other KAR channels. So this is the KAR 7.1 is the only one which has a rubidium selectivity. Whereas with the other ones, they are more potassium selective. So that's why when I used KAR 2.1, I used potassium as a preferred ion. Whereas here I use rubidium as a preferred ion. So depending on which channel I am studying, I can tweak.
But the mutation and pathogenic mutation cause disruption in the movement of the potassium.
Correct. In the channel. But then, rubidium again not physiological. So we can study it at the channel level to show whether the channel is functioning or not. But it's not a physiological ion where we would see a therapeutic outcome. We still have to have potassium to move into the channel. Okay.
So with the base editing, like showed that some mutations you were getting maybe have negative effects. So do you think you're getting a mosaic of different these and these, and then interacting or like interfering with your rescue?
Yes. So I didn't talk about, I mean, we have done several studies since we had access to these. So of these, so since we are interested in care 7.1, and then there are several phenotypes. So if, for example, here, so we have also, so this patient, as I said, this is also one mutation we are writing. And then the reason we got interested, because this is a novel mutation that we found in patients, plus we wanted to do base editing exactly after the success of W53X. And we thought with the W53X, it's hard because it is on the N terminal, and probably we are not getting a readout. Whereas here, you already have two transmembrane. So even if I insert any amino acid here, that might not be that deleterious compared to the W53X. With that intent, we tried it. But then this is not a target. You know, we can't design a guide RNA to target the L144P. So that's where we are getting into prime editing. So showing in the paper that, okay, yeah, this is not a editable target with the existing base editor, but probably prime editor is way to go for this, or gene therapy, which in our opinion.
tRNA. So of course, like you give the tRNA and there's going to be competition, right? But at some point that becomes steady state. Right? Is there like late term? So like past 8 weeks, is there an issue with competition?
So what we did with the tRNA, initially when we were trying tRNA, so we had to titrate. So because it is, as I said, it's only 70 nucleotides. So right now our therapeutic molecule is at least 3x or more. So we can package 3 of those tRNA. Okay, so with one, we do see a therapeutic outcome, but not up to the level of where we want it. So we can package 3 of those. So 3x or more. So our clinical one that we are making is now is a 6x tRNA. With that, I mean, we have a robust expression of the tRNA, continued expression of the tRNA, and then also the read-through is much better. Correct, correct. And then with the tRNA, the other unique thing about the tRNA is it doesn't use the normal promoters. It has its own promoter. So that's why, you know, I mean, so it's a small promoter plus a small gene. We can package this the way we want, and then it does the trick.
Fascinating approach to tRNA. I want to follow up with what Josh said. Are you unearthing other proteins that have probably been silenced for good reason?
Yes.
Have you done any mass spec to see whether or not there are any other proteins that you're digging out that should be buried?
Yes, exactly. So we are right now, in fact, doing mass spec. So what we found, I mean, we were initially interested in KR 7.1 to do mass spec, but then the sites that we are looking at W53X, it's not a good site for mass spec. Even for read through also, when I was testing the drugs, that was my hypothesis. If we do mass spec, probably we'll see different amino acids getting inserted after the therapy. But W53X, you cannot read with mass spec because it gets digested by the enzymes. So that's when now we are relying on other proteins and other protein screening to use the mass spec to find out, or even GFP, because we have developed GFP reporters with multiple stop codons. So we are also doing mass spec on the GFP to show that, yes, it's basically the wild type amino acid which is getting inserted, and it's not affecting any other amino acid in the sequence.
Cell, I mean, certain points.
So it's not just, yeah. So it's not just too much of the tRNA. It's in fact, even if, let's say we have tested it, even if we deliver it to the subretinal space, it goes to photoreceptor. It doesn't do anything. You know, it sits there. It's been shown for the other groups, those who are doing neuronal delivery in the CNS. So although it goes to other cell type, it doesn't have any phenotypic effect. So that's why probably we are lucky enough that it doesn't have, it passes out the safety study. Yeah.
You can ask them later then. Okay. Well, I think this is a very fascinating talk. I want to thank you very much. And I think a lot of interesting ideas that I hope will be an interest to a whole variety of people here. So thank you very much.